Hello, and then Edward, right? All right. All right, so today we're going to be is kind of the meatiest day because we're going to be talking about building complex simulations in SLIM. So I'm going to go through a couple of different cases and explain how you might build a more complicated evolutionary scenario than what we looked at yesterday. So we're going to start by talking about demography and population structure. So how might you change population sizes or have multiple subpopulations? Then we're going to talk about modeling selection in SLIM and spend our time thinking about a bunch of different kinds of selective scenarios. So we'll go through mutation types and genomic elements again. Then we'll talk about types of selective sweeps and how we might simulate those different kinds, and then context-dependent selection. I'm also going to do a quick recap, also based on what you all said during your exit tickets yesterday you were confused about. So as a recap, population genetics is the study of how genes spread through populations over time. Models can be forward in time like SLIM, where we simulate generations in a row, or backwards in time, where we take some level of genetic variation and go back in time until all individuals share a common ancestor. A forward in time model like SLIM tends to use Wright-Fisher reproduction, which simulates discrete generations, so non-overlapping generations of reproduction using binomial sampling, as opposed to other models of reproduction might include overlapping generations, where it's possible, like in humankind, it is possible for your grandmother to reproduce at the same time as your mother, given how wide reproductive ages can be and depending on when they started actively reproducing, so to speak. But in general, Wright-Fisher reproduction simulates these discrete generations. After you've reproduced, you're dead to us. And it also uses binomial sampling using expected fitnesses to figure out what the population in the next generation will look like. SLIM is a forward in time, genetically explicit architecture that makes writing custom simulations easier, but it's not the only way to do it. You can build things from scratch. It's written with this custom EDOS language, and we all, I think, decided together that it's based on Java yesterday. And so if you know Java, it's a little bit easier. A basic SLIM script boils down to 3 parts. Initialize to define all of your parameters at the start, early and late events happening during specified generations. And those happen either before reproduction occurs or after reproduction occurs, but still within a specific generation. And the SLIM GUI allows for some quick scripting, debugging, and some very limited visualizations, but it's still a powerful tool when you're getting started. And we're going to be doing that today. So to briefly recap what the Wright-Fisher model and SLIM is, because I got a couple of questions about this. So in a Wright-Fisher model, you have every individual has, especially one with SLIM, every individual has 2 chromosomes, right? One from mom, one from dad. And then in the following generation, we use binomial sampling, so a random number generator, to determine whose chromosomes you get. So let's say having a purple chromosome makes you have a way higher fitness than having a white chromosome. That means that when this individual is choosing one of its parents' chromosomes, it's going to pick this one with a probability of 1, 2, 3, 4, 5. So if all of them was neutral, the probability would be 1/5 or 20%. Let's say that being purple gives you a slight selective advantage. So instead, we're going to pick a purple chromosome with probability 30%, and the others with 70 divided by 4, I guess. So it picks the first chromosome from one parent, and then it picks the second chromosome from the other parent, using the same kind of fitness language, although it's probably, depending on the parameter inner workings of SLIM, I think the default is that it won't pick from the same exact parent. It's going to have to pick from a different parent. You can instead set it up to do other things, but it'll pick the second chromosome using that same weighted random number generator, where it has a higher probability of picking a chromosome, the higher fitness that chromosome. So then all of the offspring get their genotypes from their parents using this random number generator. And then you have genome modification events occur. And so genome modification events are like mutation and recombination. And in both of these cases, some of the slim parameters you have are a mutation rate and a recombination rate. When you initialize, what your mutation rate is it's saying for every single base pair, every single location in the genome, what is the probability of changing? So let's do some back of the napkin math. If our mutation rate is 10 to the negative 6, and you're simulating a chromosome that is, let's say, 10 to the third, and a population of a size 10 to the third, how many mutations would you expect to see in a single generation? Yeah. So mu is mutation rate. Yeah, so in a single generation, how many mutations will you get on average? Or how would we just begin to calculate it? Yeah, so if there are 10 to the third individuals in the population, and each one has 10 to the third base pairs, how many loci are there in a given generation? And if each base pair individually has a probability of getting a mutation event of 10 to the negative 6, how many, Yeah. So you would just multiply it again. Okay, does that kind of make sense for how mutation rates work? So in SLIM, recombination events are working the same way, but instead of having a single base pair that goes from a 0 to a 1, because remember in SLIM, it doesn't actually care whether it's an A, a T, a C, or a G, it's just doing 0 to 1 or back again. For recombination, it's the probability that a recombination breakpoint occurs. So a recombination breakpoint is if you have a chromosome that looks like this and a chromosome that looks like this. I'm going along, and then I'm rolling the dice, and I get whatever number that says it's 10 to the negative 6 time. I'm going to create a break here and switch these over. So now I have one chromosome that looks like this, and one chromosome that looks like this. And so why might that be important? These should all be in line. Why is that? Why do we care about recombination events? So, yeah. Anyone want to share what you think why might we care about recombination? Sure. Well, so if you have one mutation here, and let's say one mutation here, and then they switch over here and here, they're at the same, recombination doesn't change their frequency at all. Yeah. Though in the way that mutation would change the frequency of mutations or would introduce new ones. Exactly. So yeah, so this is breaking apart. So if you have two mutations that are on the same chromosome, if no recombination occurs, you're going to inherit the entire haplotype every single time. And so these will be perfectly linked, be perfectly linked. But if recombination occurs, all of a sudden, in a later generation, I might only get one of the two mutations and not the other one. And if you have weird genetic interactions, that might end up being important, or there's a lot of reasons why that might be important. But from SLIM's perspective, again, like I said, what it's doing is it's going along the base pair, it's going along every single base pair and saying, am I going to have a breakpoint here? And if I do have a breakpoint, I'm going to swap everything afterwards to the other chromosome. So using the same math, if our recombination rate is 10 to the negative 6, we would expect one recombination event per generation. So somewhere along the way. Hello, Max. Okay, I'm going to go through this again today in even more depth. But to go through different genomic elements, initialized mutation type is needed to create different types of mutations. And everything follows this naming convention of M1, M2, M3. Technically, you could have M1 and M2 both have the exact same selective coefficient, but SLIM would track them as different types of mutations. And I think in one of the recipes, it does that because it wants to keep track of like neutral mutations that are in non-coding regions versus neutral mutations that are in coding regions. You can also specify the dominance coefficient, fitness function, and mean selection coefficient. Initialized genomic elements say where mutations are allowed to go. So inside initialized genomic element, you'll say, I want to let M1 and M2 mutations, but not M3, and this is their relative ratio. Initialized genomic element, instead of initialized genomic element type, maps where the genomic element types are going to go on the genome. So we might say that in G1, you can have every kind of mutation and the entire chromosome is G1. Or you could say that G1 and G2 are slightly different, and G1 is going to be these segments, and G2 is going to be different segments. Inside of initialized genomic element, you need to specify the type, starting position, and ending position. Initialized recombination rate gives you the probability that a recombination crossing over event will occur. And you can give a flat probability. So if I say the recombination rate is 10 to the negative 6, it's going to be 10 to the negative 6 for the whole genome. If I instead want recombination hotspots, I might say that the recombination rate is 10 to the negative 6 from position 0 to 100 and 10 to the negative 3 in positions 101 to 200. And initialized mutation rate, works the exact same way, where you can either give it a flat probability or chunk it up. Finally, I think, when you're reading an SFS, so we plotted a bunch of SFSs yesterday, and it's a really powerful tool for debugging your simulations. So I want to make sure we all know how to read this. Can anyone share what the x-axis is telling us here? Mutation frequency, yeah. And the y-axis is sharing proportion of mutations. So what this has nothing to, this is a snapshot at a single point in time. This doesn't really have anything to do with generations. Just like I said, it's a snapshot in a single unit of time. These are different mutation frequencies. What this is telling us is that 75% of mutations are at a frequency between 0.01 and 0.1. Or whatever, I think the year n is not 100, so that's why I say 0.01. But whatever 1 over n is, the lowest number. Whereas mutations that are at a frequency of 80 to 90%, there's maybe only 1 or 2. This is generally the shape we expect to see in SFS, especially when there's no beneficial selection. Because mutations are one mutation is showing up every single generation, theoretically, or at least with these particular parameters. But whether or not they stick around is another story. And whether they actually spread, again, is another story. Especially if they're deleterious, they're probably going to be gotten rid of pretty quickly. Or if they're neutral. I think we played around with this yesterday. But the probability that a neutral mutation is lost in the first generation is around 1/3, depending on what your population population sizes. As you increase population size, your SFS is going to get smoother because we're doing this kind of like law of large numbers. More sampling means less impact of stochasticity. So if you look at, say, this thing here, where what percentage of mutations are at a frequency of 75%, 70 to 80%, it's like a pretty large percentage. But here, very few mutations have arisen to that frequency, or a relatively few, or a smaller proportion have when n is larger, following a less variable shape. This is expected to be like approximately Poisson distributed, but I don't actually care if you know that. I don't think I even understand what Poisson distributions are entirely. Okay, so that's it from yesterday. Today, we're going to re-talk about a lot of those things that we mentioned yesterday and as to how we can adapt and change them to make our simulations more complex. And we're going to start with demography and population structure. So why does modeling demography matter? I'm going to ask that this clump of people who all know each other, know you all know a little bit about Slim, and this chunk of people who don't, move around, change positions, break up these clumps, and maybe talk to a different person than you talked to yesterday, not where you sat yesterday, and talk for like a minute about why you think modeling different demographies matters. Yeah. I don't know. Yeah. Introduce yourselves too. Yeah. Okay. Does someone want to tell me what your group talked about? Why does modeling demography matter? This group. So somebody tell me, why does modeling demography matter? Or why do you think? There's a lot of right answers. Yeah, so if you have populations that are separated by time, you have separate demographies, they're going to just either by random chance or selection might get different levels of genetic diversity. Kathy in the chat mentioned that factoring demographic sources might include founder effects, where like if you're founding with a really small population tied to a specific region, that might be important. What did your group talk about? Yeah. So, and another thing that we already talked about a lot is that small versus big populations, demography also refers to just population sizes. And so small versus large effective population sizes are going to impact demography differently. So this is a graph from a recent paper, Cousins 2025, that shows the demographic history of humans. So A are modern humans. B is, I believe it's like an extinct early hominid ghost lineage that like when we include them, everything makes sense, but we don't actually have any genomes that represent them. And so these graphs are showing introgression or gene flow between this like ghost B population and the A population. And then the A population splits off into the Khoisan and West Africans. And then the West Africans split off into all non-Africans. And there is a really small founding population and it expands out. Then over here, there's Neanderthals and Denisovans split off from early humans a little bit later than we split off from the ghost B lineage. And there was some introgression or gene sharing between Neanderthals and Denisovans and non-Africans. 70 K. Anyway, I'm going to use the term introgression a lot because it's fun to say and it's accurate. Introgression refers to gene sharing between multiple populations. You can range between saying introgression is happening between just two populations of individuals. So you might say that like colonization of the Americas represented genetic introgression between Europeans, native indigenous peoples of America, and African populations. You can also have it refer to completely different species, like humans and Neanderthals. Or there's some really fun examples when you expand your mind and don't think about humans. So like plants do some really weird introgression, fish do some really weird introgression. Introgression is incredibly common across the tree of life. It refers to gene sharing. There's like a whole complicated thing where it's not introgression unless you like back cross back into the population. But gene sharing is a very technical way of just saying that someone from a different population, whether that is like a population or a species, donated some genetic material through reproductive processes. So anyway, we might want to think about how population expansions and contractions have impacted human diversity. We started with a small population and then we expanded out into a larger one. And how have migration and gene sharing between distinct populations been important? We've talked about this a lot, but I'm going to continue hammering it home. Population impact size impacts the efficiency of selection. And the word we use to describe the effect of random reproductive chance is drift. Some papers, it's not super common, but some papers will call selection draft. The idea, like to make it rhyme, drift and draft, where draft is like a directional change and drift is change due to random chance. But I just took a random like binomial number generator and did 10 samples versus 100 samples. Same 50% probability, and I get these two different distributions. They have the same mean, but the variance is different when you decrease the sample size. If you look at, say, like what's the probability that I get exactly 2 successes, or around even just like like around 20% successes. Here it's 0.06, which is actually a really large number. And here it's way less than 0.01. So again, at really small population sizes, the effect of random chance is a lot higher, and that's going to have really important impacts on diversity, as well as the efficiency of selection. So if you have a beneficial mutation, it's much more likely to randomly be lost in a small population compared to a larger one. Or if you have a deleterious variant, it's much more likely to randomly reach high frequencies in a small population. And in these ways, historical changes to population size can impact diversity. So at large population sizes, more efficient selection, generally more diversity. And if you have a sudden contraction, it's often called a bottleneck. People also refer to these as founder effects. But the idea is that if you started with a lot of diversity and then you go shrink to a really small founding population, and then you expand out, the genetic diversity of that founding population is going to be really important, and will lead to limited diversity or maybe even like fixing of deleterious alleles. So anyone remember what fixation means? I use this term a lot. And I just remembered that I didn't redefine it. Yeah, just. Yeah, so fixation is a frequency of 100%. Perfect. So one of the ways we assess genetic diversity is using a term called heterozygosity. Heterozygosity is a measure of genetic diversity that uses the number of heterozygotes in a population. So I've got two populations here, one that has no diversity, everyone is homozygous for a blue allele, and one that is heterozygous, or that has a lot of diversity. There's some blue allele, there's some pink allele, and there's some heterozygotes. Has anyone, raise your hand if you've heard of Hardy-Weinberg equilibrium? Great. If you haven't, I'm going to make us derive it and do it by hand today anyway. So the idea with Hardy-Weinberg equilibrium is that under a completely neutral model, if there were an infinite number of individuals, if there was absolutely no selection, if there was like no crazy mating stuff, how many heterozygotes would you expect? How many individuals with one version of pink allele on one on the mom's chromosome and the blue allele on their dad's chromosome would you expect? And so a neutrally evolving, well-mixed diploid population should have, all right, so we're going to say that P is the frequency of the big A allele or the blue, and little a is the frequency of the pink allele. If you were to take the whole population and count all of these up. So here, in this particular case, P is equal to 1 and Q is equal to 0. And in this case, there's 2, 4, 6, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 out of, I think it's 40, if I did my math right. But P is equal to 19 over 40, and Q would be equal to 21 over 40. Someone can check my math later. And so if we were to just like say our expectation, the probability that you have one of these two alleles at one locus is P plus Q. The probability that you have them at the other locus is also P plus Q, or on your other chromosome is P plus Q. So the probability that the whole probability is going to be P squared plus 2PQ plus Q squared should equal 1. P squared refers to people who are homozygous for the blue allele. So the probability that you have a blue allele at both loci is P times P. And the opposite is true for Q, for the pinkish allele, where it's Q, probability that you have Q here, the probability that you have Q here. And then the probability that you have one of each is P plus Q plus P plus P, or 2PQ. So this is like basic quadratic formula. Do you know how to do algebra? And so 2PQ is our expected frequency of the heterozygous big A, little a. And if this number is very high, then that means we have a lot of diversity because we have an even mix of both P and Q. And if that number is 0, because Q is equal to 0, there's very little diversity. So in this particular case, I want to say it's 19 over 20, or over 40, times 21 over 40 times 2, which if we round things a little bit, it should be a half-ish times a half times 2, which equals around correct? I think I can do that mental math. Right? So 0.5 times 2 is 1, times a half is, yeah, is a half. I was doing a half times a half is 0.25 times 2. Yeah. Wow. PEMDAS in action. This is why they gave me a doctorate. Okay, so this number can go from like 0 to, I think a half is the highest it can possibly be. No, that's not true. Just kidding. So, okay, what would happen if we had a really uneven mix, but it's not exactly 0? So let's say it's 7.75 times 0.25 times 2. What does that equal to? It's a half of 0.75, which is less than a half. Yeah. So the highest heterozygosity possible is 0.5. And this number will be, I don't know, someone do it on your calculator really quick. 0.4? Yeah, that makes it should be like 0.375, right? Yeah. Okay. But so the idea is that a heterozygosity of 0 means no diversity, and a heterozygosity of 5 means very high diversity. And we'll return to this concept in a couple of slides. But this is just a way of saying, like, are there more than one version of this allele present in the population? And are they at similar frequencies? Okay. So demography just refers to things surrounding population size and movement. And so it can refer to instantaneous population size changes like this one. So let's say we wanted to simulate a large population size and then a bottleneck for several generations, and then it expands back out into a larger population size. This is really easy to do in SLAM. So if you'll recall from yesterday, the first thing you do after the initialized section is always during one early, you add a subpopulation of a certain size. At any point later, you can just change subpopulation size. So we're taking the P1 object and we're modifying it. So P1 dot set subpopulation size to a brand new number. And it'll stay at that number until you change it again. So at a later generation, I'm going to change it again to a bigger number. That's really all you need to do to change population sizes instantaneously. And then I finish the simulation. If you want to continuously change population sizes, it's also very easy. But we're going to now change the population size every single generation. But we're going to do it using an equation rather than manually changing it every generation, because that would be annoying. So we started by setting the P1's population size to 100 individuals. But then from generations 100 to 1,000, we're going to do an expansion where the population size is gradually increasing every generation. And we'll use an equation that just says, what's the old count times 1.03? So this will give you a gradual exponential growth. But you could also do a linear growth where you're multiplying the current generation by some constant or some slope. Or you could do really any equation you'd like there. But bam, now you have P1 set subpopulation size to new size. And if you wanted to do, you can also introduce a cap. So let's say I want to set a carrying capacity of 10,000. Now I can say if new size is greater than 10,000, I'm going to reset it to 10,000 and bam, you're done. And then you can also set the equation to be context dependent, maybe based on mean fitness. So there are examples where you might want to say that, like, if the population's mean fitness is gradually decreasing due to an accumulation of deleterious mutations, you want the population to go extinct. And so you would do so with something like this, where you're saying the new size is 100 times the mean fitness of the population. And I did not actually quote out a contraction here, but a contraction, you would just multiply this by a number smaller than 1, and it would slowly get smaller at a rate. That's fucked up. Okay, that's it on like changing population sizes. I'm now going to talk about subdivision and migration. So what happens if you want to simulate more than one population at a time? Migration impacts how well connected two populations are. So let's say that I have the blue actually came from one population and the pink came from a different population of individuals, and I allowed them to meet. And many generations pass. And in one case, they stayed distinct. Maybe there is a giant mountain range in the middle of these two bird species, and so they're not actually meeting and there can't actually be gene flow. Or maybe there's tons of gene flow, there's tons of migration between population 1 and population 2, and they are actually very well mixed. So how can we can see this very visually here, but how might you mathematically quantify it? So we're going to return to Hardy-Weinberg to do that. The FST is a common population genetics measurement of how distinct populations are. And it leverages this Hardy-Weinberg expected heterozygosity to tell us whether or not populations are distinct or well mixed. So FST stands for the fixation index, and it's going to be the expected heterozygosity minus the expected heterozygosity minus the mean expected heterozygosity for each subpopulation divided by H total expected. Okay, so in this case, what is HT? What is the total heterozygosity? What would we expect heterozygosity to be? Well, exactly half are blue and exactly half are pink. So HT is equal to 2 times a half, which we've already established is a half. Now, and the same is true here. So now, what is the heterozygosity of this individual population? Or what are our expected heterozygosity of this population? If we were to ignore this side, what would this heterozygosity be? 0. And this one also 0. Now, what is our heterozygosity here? 5. I've already shown you the answer. It's exactly a half, or it's very close to a half. It's 10 out of 20 times 10 out of 20. And it's also true for population 2. So FST, or the population fixation index, for this example, is exactly 0, because it's half minus a half. And on the other side, H1 is 0, H2 is 0, so our FST is 1. So very low values of FST mean that there's the heterozygosity we would expect to see in the entire population can also be observed in the subpopulations. Or if I take an individual subpopulation, its level of diversity is indicative of the entire metapopulation is a fancy term for it, but the overall combination of all subpopulations. Whereas if SOST is equal to 1, that means the two populations are very different. And if I were to take one individual population or subpopulation, that won't tell me a lot about the other subpopulation. SLIM has a built-in calc FST that we're going to play with very shortly. So you don't actually have to do this. I also want to highlight really quickly that FST is actually not calculating the realized heterozygosity. It is only using allele frequencies to calculate the expected heterozygosity. So what you might notice is that here, like heterozygosity, the real heterozygosity isn't exactly a half. That's okay, it doesn't actually care about that. There are other statistics I can talk about later that can tell us whether or not selection is going on based on a mismatch between expected heterozygosity and realized heterozygosity. Okay, so another aspect of demography might be having multiple subpopulations. So if I wanted to code up this complicated scenario where I have two subpopulations to start out with, I would say sim add subpop 1, P1 equals 1,000, and add subpop P2 equals 1,000. Your populations follow the same naming convention as everything else, where you have to start with P1 and then go to P2 and P3. You cannot change that. You can also just get rid of subpopulations at any point in time. So let's say I want to have P2 go extinct. You just set that subpopulation size to 0, and that's the way you get rid of it. At any time, you can split a subpopulation. So I might say, sim add subpop split. P3 is saying the new name of the subpopulation. 750 is saying how many individuals should I take from P1 to found P3. And P1 is saying the source. You can also give it a vector, 2 vectors here. So let's say I actually wanted P3 to be founded half from P1 and half from P2. I would say, 300, C 300, comma 300, P1, comma P2. The vectors here. Whoops. And to go back, P1 in this case is just going to be 1,000 minus 750. So any individuals that are left will go down. You can also specify migration and gene flow in SLIM. So using a similar kind of scenario, when we create these populations, we can also set migration rates. SLIM uses immigration rates into a population. So if I say P2 set migration rates, I am saying that the migration rate into P2 from P1 is 0.2. What SLIM is doing here is saying that with a 20% chance in the next every generation, I'm going to choose my parent from one of the genotypes that originated in P1. Does that make sense? Because we're not actually tracking migration like you would kind of imagine it. It's just where you're allowed to pull the parents from. That also means that there can be stochasticity in the realized amount of migration or amount of gene flow. Because you're setting immigration rates separately for every single population, they can be very one-sided. So if in this particular case, I've set a migration rate for P2, but I haven't set one into P1. So that means that for individuals in P2, there's a 20% chance your parent is actually from P1. For individuals in P1, there's a 0% chance they ever take your parent from P2. Then I did the same kind of add subpop splits. Again, you can give a vector of multiple migration rates. They need to definitely add up to less than 1, otherwise it will break. Because these are probabilities that you pull a parent, right? So here I'm setting migration rates into P2 from both P1 and P3 and saying there's a very low probability, but a non-zero probability I pull your parent from P1 and a larger probability I pull your parent from P3. And then I did the same for P1 and P3 just to make it look like the picture. And so P1 and P3 have symmetrical migration rates, but I specified it separately. Okay, that was a lot of information. And so I'm now going to have you all practice doing this. You can do groups again. People in Zoom, I'll put you in a breakout room so you can talk to each other without everyone in the room hearing you. Whoops. Let me go back a bit. Okay, so you're going to practice modeling the demographic history of kids. I've done some weird scaling. I'll talk about this later. If I basically divided everything to 10 to make it run on your computers really fast. This is a zoom in of a much larger figure with like way more complicated demography from a paper by Chavez in 2019 that looked at the demographic history of canids. So wolves, domestic dogs, and coyotes all shared a common ancestor that had a population size of around 1,764. You can round, just as long as you get in the ballpark of these figures, that's good. And then it split off around 10 to the 3 generations into coyotes and the common ancestor of gray wolves and domestic dogs. And then gray wolves and domestic dogs broke apart from their common ancestor a lot more recently. So here are a bunch of population sizes and migration rates. I want you to note that there's migration into coyotes from lupus to latrans and from familiaris to latrans, but not the other way around. It's very asymmetrical. So given all of these parameters, I want you to create a slim code that does this. You don't necessarily need to have multiple mutation types, although you can if you want. You can use any recipes you want. There's only 3 populations to keep track of. I'll give you like 10 or 15 minutes to do this, and then we'll come back together and talk about it as a group. You're welcome to work together in your small groups of 2 and 3. After this activity, I'm going to make you switch again. People in person. There's so few of you. I feel like I can do anything. Yeah, okay. Anyway, spend 10, 15 minutes getting this to run. And then what I'd love you to do is tell me which populations at the end of your simulations had the highest diversity and which pairs are the most similar. So I don't think I told you how to do that. At the end here, so after you run your simulations, you can open up a command line thing and use calc FST and calc heterozygosity. And I will write down how to do that on the board, or I will actually find it in the manual. The manual is so easy to find stuff in. But calc FST, you can only do it pairwise, so you're going to have to do calc FST 3 times to get all of the permutations, just as a small note. But calc FST is very easy, and calc heterozygote is all very easy. Anyway, spend 10 minutes. I'm going to put you guys in a breakout room. We're on Zoom. And yeah, take 10 minutes to just get in there. And I'm going to go back so that I'm not covering things up. I believe that's the syntax. Correct me if I'm wrong. It's in a slide that's way later. I don't know what I was on. Yeah, assume, yeah, do not try to do anything. Yeah, yeah. Yeah, so you can assume that if I say that the NE for coyotes here is 657, here the NE for coyotes is also 600. That's kind of what I'm, yeah, that's what I'm trying to tell you. Yeah, and just so you know, 657 and 1243 do not add up to exactly 17. No, so the original population is there, and it'll be the rest of the population. But you can in the kind of the right way or the way I like to do this is you can reset that population. So if you do a subpopulation split, you can also the original population would be there, and you can set its population size to whatever you want it to be, just to like. Yeah, exactly. Or the yeah, and this is the to like be very like persnickety about language. This is the common ancestor of rainbows and dogs. This population. There's like, I mean, there's a ton of different ways you can do this. I think you could call by the end of the simulation, any of these could be called P1, 2, 3. What does it actually matter? As long as you know which one is your coyotes, which one is your gray wolves, which one are your domesticated dogs. Also, I do not think these introgression migration rates are accurate. My last job was looking at canid genomics, and there is so much more gene sharing than that. There is so much more introgression. That's a really good question. That's also, I don't think is true. So, well, okay. Amongst domestic dogs, the domestic dog, well, I mean, okay. So when we talk about introgression rates, this is like getting really into it. If you have a mommy Canis familiaris and a daddy coyote, you end up with a hybrid. That's not introgression, because that hybrid could just be a single individual. If that hybrid then goes back to the source population, you end up what's called with an F1 cross, which is three quarters one population and 1/4 the other population, and that's introgression. That's like a very like moot point. But anyway, the point is that you have an initial gene flow, an initial event where you introduce some amount of dog genetics in, and then it gets brought back into and like gets incorporated into the coyote population. I have no idea why this paper. I didn't read it super carefully. I was just looking at for photos that looked nice. It had population and yeah, I don't know why their migration rates were so one-sided. My guess. Oh, I had a friend in grad school who researched like sex biased migration, and found that there's oftentimes a lot of sex bias in migration rates or in like these introgression events, where like it's very common for like it's always. I mean, you can kind of imagine just based on like male and female biology that if it's one-sided, it's yeah. So it could be different number, but like also individuals tend to be raised by the birthing parent, and so you tend to get back crossed into your mother's population. Is like one has been a is like a really interesting area of work, and they use like amount of introgression on the X chromosome versus the Y chromosome and so on to look at that. Yeah, I have no idea why their migration, why they got those, or how they got those, or I do know how they got those migration rates. They were looking at levels of variation. One thing you'll note is that the coyote population is a lot larger than either the gray wolf and the domestic dog population sizes. And so there's just going to happen to be a lot more diversity. When your group finishes, or when you, maybe everyone can run it. I'm going to make a table. When you actually finish coding this up, and people who are zooming in, if you want to send it to me in the chat, once you get a heterozygosity score for the end of your simulation for each of these, come up and write it down, and then we'll do once you calculate FST between once you finish, and so again, I want you to calculate heterozygosity for each of these population sizes independently, and you can do that just using the command line interface. And then I want you to calculate FST for every pair of populations. And when you finish, come on up and give me, feel free to write down. And people who are zooming in, if you could send it to me via chat, that would be great. The migration rates only start after 10 Okay, so this is going to create so this is going to calculate yeah. So so that's yeah, yeah. Yeah, so I would do so how about us use a pairwise fix? Unfortunately, thank you for you might want to, so I'm not going to ask you to upload this at any time. You might want to save it for your own reference. There is a question on the exit ticket about it, but if you pay attention during, like, you might not need it later. It's p1.genomes. Oh, so is it haplosomes in some time? I haven't actually updated to some which I probably should have done before we had a workshop. I think will genomes not work anymore? All right, we'll try it out. Yeah, so you can also, another way you can do this is you can click on after you can find and just you have to do it 3 separate. I know it's annoying, but so FST is a metric of like pairwise heterozygosity. Well, if you have SLIM4, it's genomes, and if you have SLIM5, it's haplosomes, I guess.
Oh, I mean, you can read it. I don't know if
And so I do not care if you say that coyotes are P1 or coyotes are P1.
because I at P for the coyote is supposed to be. Yeah, so when I do P2 add population split and do 657, for me P2 is the coyote.
Yeah, that's fine. I do, as long as you keep track. So you're doing, so you guys are doing the exact same thing. So the color triggers are this. If all of the, you should see.
Yeah, I should see that. It's 4.
How do you see that?
Project.
They're not.
Oh, I haven't changed. Oh, were we keeping the mutation?
Am I writing this in for you?
Make it whatever you want, but his mutation rate is 10 times higher than yours. Okay, this is what,
Like haplosomes is only slim. It's like never nowhere else to use.
yeah. Yeah. And so because you all have different mutation rates, you all have different like other things, and also just due to random chance, I'm not expecting you to all to get the exact same number. In fact, that's kind of the point of SLIM, is that it's a random number generator. It's a stochastic simulation. You will not get the same number every time. Yeah, it's haplosomes, not genomes. As one as well, yeah. That is a really silly thing to do.
Yeah,
Yeah, so if your mutation rate is super low, you're gonna get fewer mutations on average. So it's less opportunities to have that successful. We're mostly going to be looking at like the relative, because you've got like you're all different other parameters. Yeah. Yep. So it brings up this. I can't see. Trans
oh, sorry, like P2 would be okay.
yeah. So, okay, one thing I'm using is this is revolutionary.
Oh,
You should be doing.
oh, it should be P2 and then P1 on this.
Yes. Yeah. So remember, migration rates are into, not out of.
Oh, now they look for offers.
That's why, because you were going the wrong way.
Yeah.
Okay.
Squeeze back so we sort of put it on the student. So, yeah, so which one is what is according to this what is which one is yeah. So what is so what is and which is
Okay, I'm gonna have us discuss. I'm gonna move on and have us discuss results. And we can. So, all right, what do we see? What do we see overall for diversity scores? Yeah, using heterozygosity. Yeah, so which has the tends to have, among all of your simulations, dogs have the lowest because they also. Why might that be? Yeah. They've also gone through like very recent bottlenecks. And which population has the highest diversity? Yeah, I think that's true for everyone, right? This number is so large, like my brain can't, or there's so many zeros. But yeah, overall, coyotes have the highest, they've had the largest population size for the longest period of time. So if you were to suddenly increase dogs' population size to 657 in the last like 2 generations, you still would see similar diversity scores because it's a very recent bottleneck. Okay, what about FST scores? Which 2 populations were the most well connected? Wolf and dog. Why are wolves and dogs the most connected? Great. So we also used FST. So when we were looking at our migration rates, both of our migration rates were into the trans or coyotes. So if you look at coyote and dog and coyote and wolf, which of those 2 was the next highest? Yeah, coyote wolf. Does that match the migration rates we see? I don't think so. why is there a mismatch, do we think? Yeah, that's I mean, like, I would say that's probably the answer, is that there's like dogs are so different because they've had such a huge bottleneck founder effect. Yeah, so yeah, that's a really common, that's a good answer. Here's a trick question. Who are more closely related, coyotes and dogs or coyotes and gray wolves? Just looking at the phylogenetic tree up there. Your tree, what? Yes, that's the correct answer. You can't tell. They're both equally related. This is not a phylogenetics class, but I'm gonna get you anyway. So coyotes, gray wolves and dogs share a common ancestor, and that is who is most closely related to coyotes, but we cannot tell which one is more closely related based on the phylogenetic tree. Now, based on this, we might say we are saying that actually the dogs are slightly, or the overall wolves seem to be more closely related, but that's due to like founder effect and not necessarily reflecting phylogenetic relationships. And if you were to zoom out, that's not what you find. All right. What? They came back from their breakout room, and they did not respond in the chat. So I don't know. Zoom is hard. I can badger you guys to participate, but it's a little bit harder on Zoom. Okay. So code is still running. Well, when you get your code to finish running, feel free to add it. We're going to spend the rest of the day talking about modeling selection, but we'll give you all a 10. I know we just had a breakout group, and so some of you probably use the restroom, but if you want to take a quick 10-minute break, you can do that now, and then we'll come back and talk about modeling selection. Today's gonna be a long day. We might not finish everything in time. In which case, I don't have to make slides for tomorrow. I will try to wrap. I know there's a seminar that a lot of us want to go to. Yeah, we'll see. But the demographic stuff was supposed to be short, so we probably might not get through today. Yeah, maybe 5-minute break. All right, so we're now going to talk about modeling selection and get a little bit more in depth into how what genomic elements and mutation types look like. And we're not going to get through all of this today. Some of it's going to get, I think the most complicated section at the end is going to get punted to tomorrow, which is great. Okay, so in initialize, we've already gone through this a couple of times, but I'm going to mention it again. You create different mutation types in genomic elements. So, and we've already been through this, but they follow this M1, M2 strategy. With a gamma distribution, you have to include both the mean and the variance. And what we're saying here is the dominance coefficient, what we're naming this mutation M1, 2, 3, dominance coefficient, the distribution we're using to calculate fitness, the mean of that distribution, and for the gamma distribution, the variance. Genomic element types say where mutations can go. And for a given genomic element type, you can give it a vector of mutation types and their relative proportions. These relative proportions do not have to be fractions, or they can be integers or floats. It doesn't super duper matter. It's just going to do a ratio. So in this case, I'm saying that in the G1 element, the most common type of mutation types are M2s, which are deleterious. So M2 mutations occur 4 times as often as neutral mutations, and beneficial mutations occur very much less frequently. They appear at 1/100th of the rate of all other mutations combined. If I did quick math correctly. I might say the genomic element type G2 is an intron. This is adapted from a slim cookbook recipe. So I'm saying that genomic element type 1 is exons, which are kind of the coding regions of your genome, and most mutations are deleterious. And then I might say that in the G2 region is your introns and no beneficial. Just kidding, that's a lie because M3 are beneficial. No deleterious mutations are allowed in the introns, I guess, in the simulation I wrote, which is not how that works. And then in the non-coding regions, you might say that only neutral mutations are allowed. And then finally, initialized genomic element, not genomic element type, makes a map of where each element is. So I'm saying G3 is going from 0 to 100, G2 is going from 101 to 150, G1 is going from 151 to 250, and G3 is going from 250 to 100. So you can see this map I made. This is the slim GUI itself, but this is what each genomic element is. So you can see that the G3 elements are dark blue, and they're at the beginning and G2 and G3. Any questions still on this? This is like kind of the most important part, and I want to make sure that we're not confused. Give me a thumbs up if this makes sense. Great. Okay. As a side note, the main reason Slim uses these M1, P1, G1 naming conventions is so that you can use for loops. So for example, let's say I was making 10 different elements. I might make a for loop that looks something like this. For index 1 to 10, initialize genomic element. Oh, just kidding. This is not the right thing at all. This is creating a genomic element. This is totally the wrong thing. Whoops. Okay, so in Slim, you can use for loops to create multiple subpopulations. So let's say I wanted to create 100 subpopulations. I do not want to say P1 dot add or sim dot add subpop P1, sim dot add subpop P2. So I can instead do a for loop where I say for index in 1 to 100, sim dot add subpop. And then you can just write index and it will call it P1, P2. You don't have to like do crazy appending formats. I will fix this after class today so that when you look back at these slides, the slide is correct. But that's why it's using this like naming convention where it has to be numbers and they have to be in order. I also want to highlight a couple of Slim's mutation tracking hacks that it uses. I already talked about one, which is converting to substitutions. So if you look at this simulation, you'll notice that like sometimes a beneficial mutation shows up, it reaches high frequency and then it disappears. That's the converting to substitution hack in action. I'll play this one more time for you. So you can see, and then it disappears. So when it reaches a frequency of 100%, Slim is going to just delete the mutation because it's fixed. And so it's not going to actually have any fitness differences. It's not going to cause fitness differences within the population. That means that it has fewer mutations to keep track of, and so it can be faster. But if you want to not have Slim do that, maybe you want to allow fixed mutations to stick around because you're thinking about complex genetic interactions. You can say M1 to take the M1 object and turn off converting to substitutions. And you do that inside of the initialize block. The other mutation tracking hack that I want to highlight is called mutation stacking. In Slim, by default, if I get a mutation at locus 4, and then later I get a mutation again at locus 4, Slim lets me have both of them. In Slim's opinion, or like in the maker of Slim's opinion, this is essentially the same thing as just having two mutations that are right next to each other. Because if you have two mutations that are right next to each other, they're going to be really super duper tightly linked. And it will be very unlikely for recombination to break them apart ever. And so it's like, it's kind of the same thing, so I'm just not going to worry about it. But you might want to have a model where you allow for back mutation, where if I can get M1 here, but sometimes it mutates back to the neutral state, in which case you can also turn it off. So here the default is S for stack, and the fitness is calculated as if you have both of them, again, like they're just next to each other. But you can set it to L for last or F for first. And inside initialize, you would say M1 mutation stack policy last. And so in that case, it would overwrite it. I don't think that's correct, though. I believe what you have to do is like, oh, you have to like create a mutation stacking group. Yeah, I don't, this might not be right. And so that's correct. Okay. There you have to create a mutation stacking group before this, I believe, which I didn't include. I'll revisit this tomorrow. You have to say like create mutation stack group M1 and M2. And then you have to say M1 and M2 mutation stack policy equals last, I believe. I'll revisit this later. Just to keep, but again, the default is fine, but just keep it in mind in case you're doing really complicated simulations later on, or like, I think it's important to know why they made the choices they did. And so again, the main reason for this is that the probability of this occurring is relatively low for a given mutation rate. It's 10 to the negative 6 times, or it's whatever your mutation rate is squared. So it's going to be a pretty small number. But things happen, like things that happen with small numbers happen if you're doing a billion simulations. When you're constructing a selective sweep, a really common best practice is to use what's called a burn-in. So a burn-in is a way of establishing neutral variation before selection starts. And the general rule of thumb is you want to do about 10 times n number of generations. I was reading a paper that was like, actually, that's not enough. Maybe you need to do 20 times n. Depending on what you're actually doing, that would change. I think 10 times n is fine for the purposes of the class. But so you can use sim tags in Slim to turn on or off the ability to have certain mutations. So let's say mutation M3 is my beneficial mutation. And I'm going to add within mutation M3 this sim tag. So return if else sim tag equals 1, true, false. So this little mutation M3 sim tag function is just saying that if I have my sim tag equal to anything other than 1, I'm not going to let M3 mutations happen. And so after the initialize stuff, when I do the early thing, I might set sim tag equal to 0, which allows no beneficial mutations to occur. And then once the burn-in is complete, so at 10 times n, so here I'm assuming n is 1,000, I guess, I set sim tag equal to 1, and bam, now you have beneficial mutations. You can use this sim tag format to keep track of a lot of things, but this is what I primarily use it for, are these burn-ins. As like a brief theory interlude, one of the key aspects of this is the dominance coefficient, H. And we've talked about this a little bit yesterday, but when you're initializing a mutation type, you can give it a dominance coefficient that varies between 0 and 1. And just to illustrate why this matters, imagine if you are heterozygous, or if you're homozygous for the wild type little a allele, you have a fitness of 1. And if you're heterozygous if you're homozygous for the mutant big A allele, you're getting the full selection coefficient. Dominance coefficient mathematically scales how much of the selection coefficient we actually get to see by this dominance coefficient. So different values of H might look like this. If H is exactly equal to a half, they're considered codominant because the fitness of the two is of the heterozygote is exactly the mean. If it's greater than 0.5, that means that big A is dominant, the mutant is dominant. And your fitness is going to be closer to what it would be if the big A is entirely dominant. If it's less than a half, little a is dominant. Then there's weird cases called overdominance, where one allele is where being a heterozygote gives you a bigger advantage over either of the wild types. A really important example of this is malaria. So in humans, having sickle cell anemia actually helps protect against malaria. If you're homozygous for sickle cell anemia, it's bad because you have really bad sickle cell anemia. But if you're heterozygous, your sickle cell anemia isn't too bad. Your blood is still good at transporting oxygen, but you also get some protection from malaria. So that's a case of overdominance. And underdominance is kind of the exact opposite, where the heterozygote is less fit than either of the homozygotes. So another aspect of theory to keep in mind when you're parameterizing your simulations is the efficiency of selection. In general, the rule of thumb is that selection cannot see mutations that have a selection coefficient that is too close to 0. And we can use population size to figure out what that boundary is. So just to like put it in even simpler terms, if we have a super duper beneficial mutation that like doubles your fitness, it's gonna be really easy for selection to see that mutation. It's gonna fix basically all of the time. But if you have a mutation that gives you a selection coefficient that is very close to 0, it's going to be essentially neutral. And if we bring back in mind these aspects of population size and how smaller population sizes mean selection is less efficient, we get this general rule of thumb. So in your switch up your teams, send a trade. Go back to a simple simulation with 2 mutation types, neutral and beneficial, and have one person set the beneficial mutations S to 1 over n, or to a number less than 1 over n. So if you use an n of 10 to the 3, have one person set it to something, have one person set it to 10 to the negative 4, and another person set it to 10 to the negative 2. Is that, or if you're in the group of 3, have one person set it to exactly 0, one person have it set it to a little bit less than, 10 to the negative 4, and one person set it to 10 to the negative 2. Those numbers aren't exact. And then after you finish doing that, generate an SFS and compare your SFS. Remember to include a burn-in before you introduce your beneficial mutations. And switch up the teams a little bit. Yeah, you can all just work together. When I wrote these slides, I was expecting 16 people. Yeah, so what I'm looking for is burn-in and then maybe like start allowing beneficial mutations to happen and let it go for a couple of generations, or I don't know, maybe like 10 times n more generations or something. And I just want to have some people set S really large and some people set S really small and some people set S equal to 0 and compare those SFSs. The goal, well, I'm going to like cut to the punchline and tell you what we should see, which is that people who set S to be less than 1 over n should have the SFS for neutral mutations and your beneficial mutations should look similar. And people who set S to more than 1 over n should see a different SFS, where the beneficial mutations are like more likely to rise to high frequency. Really small. Might not look like that. Okay, so what did you set S equal to? Yeah. All right, run it again. It's like way less than 1 over n. You do not have to. So is there, how are you planning on? Yeah, that's what yeah, you can use some tags to do it. Gotcha. Okay, so there are two ways that you can introduce beneficial mutations. So the first is you can let them happen kind of organically through mutation. So you set some mutation rate and then you let it go. The other way you can do it is to say, I'm gonna, after the burn-in is done, I'm gonna randomly take one individual and give them the mutation I want and see if it suits. So I don't think that's the right way to go for this particular case because that in that particular case, you'll only have one different type of M1 mutation or of the beneficial mutation because you only introduce it once. The only way it can increase in frequency or decrease in frequency is through reproduction. You kind of want the mutation to be like recurrently mutating in order for this particular exercise, but a different exercise where you're just like, if I introduce a brand new mutation, what is the probability of success? Where it only is introduced once. Add draw mutation is great because it means that you don't need to wait for that mutation to show up. Right. Works. Always good to make sure it's like a gravity check. Yeah. I don't care. That's not important for this. Like, I really only think I care about. Make it whatever you want. Yeah, great. Okay. What part of the theory? Okay, S is this selection. All right, so to like make a okay. So as a reminder, S is our selection coefficient that tells us how different the fitness is from the wild type. Um, so remember, S is fitness of the mutant. Oh, fitness is with a W. I don't know why. Minus fitness of the wild type divided by fitness of the wild type. So this is the relative increase in fitness that a given mutation gives you, is what S is telling us. So S can be anywhere from, technically S can be anywhere from like negative infinity to positive infinity, although in practice, most people go from -1 to 1, and 0 is in the middle. So what does 0 mean? What does having an S of 0 mean? Neutral, right? It doesn't change your fitness at all. And in general, we say any S's greater than 0 are beneficial, and any less than 0 are deleterious. Okay? What this particular thing is telling you is that actually neutral doesn't necessarily mean it needs to be exactly 0. There's kind of a fuzzy border around 0 at 1 over the population size, where mutations that are only really weakly deleterious or only really weakly beneficial are so close to neutral that they're going to act like they're neutral. And so we include this barrier of 1 over n. And the reason why it's 1 over n is because it depends on population size, because population size, the efficiency of selection is related to population size. I'll also mention that the reason it's exactly 1 over n has to do with the fact that 1 over n is the change of a single individual. Okay? So just to like, let's say you have 10 individuals and your S is equal to 0.000001. There's a single individual. That means that we expect there to be 1.00001 individuals the next generation, which if we round it to the nearest 1 is exactly 1, right? But if we instead said that S is equal to 0.5, we would get an expected number of individuals of 1.5, which is close enough to 2 that it's meaningfully different. I hope that kind of makes sense. Both. So it's a, we like to, so everything is sensitive to drift. Anything can be lost to drift, whether it has a, and actually the probability of the probability of fixation of a neutral allele is approximately equal to its starting frequency. So if it starts at a frequency of 1 over n, that's the probability of fixation, which if you're talking about 10 to the -4 probability of fixation, like probabilities of fixation that are considered indicative of being beneficial do not need to be that big. In fact, it's getting lost most of the time. It's just fixing slightly more often than we expect. Yeah, it's not super important. I mean, yeah. Yeah. So let's look at the frequency. So you're saying So that depends. So let's say, what do you want your population size? Okay. Yeah. Initialize constant, I think. I think it's initialize constant. Now we need to so let's say yeah, so if we want to do like 10 it's like 26. Yeah, so the times of so, so there's two different things. So one of them is which says like mutual. I think so. This is kind of doing I don't know. Yeah, I don't know. So that's so G1 is telling you yeah, I think
I'll give the basic.
so. Yeah, so well, first of all, we're now at the end of this. So beneficial mutations are in past. Okay, so just to this is saying every generation, every single individual gets two of these chromosomes. And in these chromosomes, these beneficial mutations are okay. That's so every individual can have, there's 360 locations for the beneficial mutation. Does that make sense? So this is a generation. At generation 1, there's generation 1. There's 10,000 minutes about 9,000. Generations make sense. Okay, I understand that they come. So that's one of the reasons why they call these types of shortens to two. So try making S, the S here really small. It's 0.00000. Yeah. Okay, I am going to call it here. Hold on. Okay, so I'm gonna say that we're gonna end here and move the more complicated selection scenarios till tomorrow to give ourselves more time. So before you leave, complete the exit tick again. Please make sure it works. I just changed it. So essentially, I'm just asking you to I'm going to erase this and make you remember it. But we did 2 main activities in class today, the demographic history one and the efficiency of selection one. And so the first question is asking about the first, and the second question is asking about the second activity. If it asks you about epistasis, it's that is it. Is that right? Yeah, okay. And then at the end, I'm asking you to upload an SFS where the first question is asking you to upload an SFS where selection is very weak or where S is a lot smaller than 1 over N. And then the second question, I'm asking for S of that's a lot bigger than 1 over N. And people in chat can here's the exit ticket as well. And as a reminder, the exit tickets are how I'm doing your homework assignments. And I am grading them for, like, I am going to actually read your responses and grade them. But they're also how I get better. So even if you're not taking the, yeah, I'm going to fail you. No. What? What did you say? Oh, okay. I was like, even if. I did erase it. Yeah. I'm making you remember things. Anyway, even if you're not taking the class for credit, completing the exit tickets helps me make sure that I know that that you've got something out of it and that you're not confused. And it helps me improve as an educator. So even if you're not doing it for credit, I would still appreciate if you could try to fill out the exit tickets, which everyone in the room did, but people on Zoom didn't. So I'm gonna end class, or I'll stay on for a little bit in case people on the chat need to ask me a quick question. But as soon as you complete your exit ticket, that is your ticket to exit. And so you can head out and attend any seminars you want. But I will also be here for the next 30 minutes to help if you need it.